Hi, my name is Ahmed Ahmed, and I'm a final year PhD student at the National University of Ireland, Galway, under the supervision of Dr Constantina Papatriantafyllopoulou. We are part of the materials team at SSPC, and I'm here to show my work on metal organic frameworks for drug delivery. So just an introduction, many drugs suffer from issues such as poor solubility, bioavailability, or they are rapidly metabolized and excreted by the body. To overcome these issues, drug carriers are required. However, there are many issues with current carriers, from low loading capacity, poor stability, and in some cases, poor bioavailability. Metal organic frameworks are crystalline porous 3D polymers composed of metal-ion clusters and bridging organic linkers. You can see a schematic on the right-hand side of a porous 3D framework. MOFs have been shown to have large drug loadings and the potential for controlled drug release, which can help overcome many of the issues of current carriers. You can see a schematic on the bottom there of how this works. You synthesize a non-toxic MOF, encapsulate your drug or biomolecule, and transfer it into the biological medium and get controlled drug release. So work to date, we work on the synthesis of new metal organic frameworks based on non-toxic metals. These are things like iron, zinc, magnesium, and calcium with carboxylate linkers, which have been shown to have low toxicity. We characterize the MOFs using X-ray diffraction, IR, thermogravimetric analysis, as well as a host of other techniques. We then studied encapsulation and release of many drugs, including anti-cancer drugs such as doxorubicin hydrochloride, which you can see the structure of on the right-hand side, 6-mercaptopurine, and other drugs such as carbamazepine. This is done using UV-Vis and HPLC. So a MOF we've recently published here is NUI-G4. You can see on the right-hand side a representation of its pore structure. It's got these large 19 angstrom pores, and we successfully encapsulated doxorubicin with a record loading of 1,955 milligrams of doxorubicin per gram of MOF. So in collaboration with many members of SSPC, including Dr. Sarah Hudson and Dr. Sarah Gurn, we showed using solid state NMR and computational studies that this large encapsulation is facilitated through pi-pi interactions between NUI-G4 and doxorubicin. This is crucial to the very high loading. We then carried out drug release studies in 3 different solutions, just pure water, phosphate buffer saline solution at 7.4 pH, as well as acetate buffer solution at 5.5 pH. We found that the release is pH dependent with greater release in aqueous acidic solutions. We then carried out biological studies to see if the framework itself is biocompatible. So we encapsulated doxorubicin and we tested that encapsulated doxorubicin versus doxorubicin itself on human dermal fibroblasts. And we found that we increased the LC50 from 3 micromolar for doxorubicin to 25 micromolar for doxorubicin encapsulated in NUI-G4, just showing the protective effect of the framework on healthy cells. So our current work includes the synthesis of functionalized variants of NUI-G4, which will allow us to hopefully control the release and toxicity of the framework itself. And we're currently working actually on MRI active MOFs, which have the potential for drug delivery and simultaneous imaging. So just to summarize, we've synthesized and characterized new non-toxic metal organic frameworks, encapsulated and studied the encapsulation and release of anti-cancer drugs under simulated physiological conditions. We then studied these interactions using solid state NMR and computational methods through collaboration with many members of SSPC. And I'd like to thank everyone down at the bottom, particularly Dr. Rana Sani, Dr. Sarah Hudson, and Dr. Sarah Gurn, and Dr. Damian Thompson for working with us on this project. Thanks very much.